Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 12th biology students, and this is for 12th September 2020. We have been discussing about biotechnological applications in the field of medicine, and we are thankful to this field for making artificial insulin which helps in human beings having diabetes, and this artificial insulin synthesized through gene, and it is called humulin. So in our last session we were discussing about the preparation of humulin by an American company in 1983, Eli Lilly, and of course we are not restricting ourselves to that. We extend this scope in biotechnology to other incurable diseases, I said, and that is called gene therapy. So by using suitable gene, we are able to cure certain incurable diseases through gene therapy. So it is a collective therapy to correct or treat hereditary diseases, a gene defect diagnosed in a child or embryo. So the method where genes are inserted into the cells or tissues to treat diseases. The principle of gene therapy is normal gene when get inserted into individual cells or embryo with the gene defect, the normal gene will take over the function of and compensate for the non-functional gene. So first clinical gene therapy was done in 1990 in a 4-year-old girl who has got ADA deficiency, adenosine deaminase deficiency. Initially it was treated through bone marrow transplantation and by giving certain enzymal injections, enzyme replacement therapy, but the results were not that good. Hence the gene therapy is resorted to. So in gene therapy, it is a collection of methods that allows genes to be inserted into a person's cells or tissues to treat a disease. Correction of genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with ADA deficiency, adenosine deaminase deficiency. This enzyme is very important for the immune system to function. So SID, that is SCID. How do you expand that? Severe, S for severe, C for combined, I for immuno or immune, D for deficiency. So that's how we say SCID. So severe combined immunodeficiency, that is SID. And this SID is a group of rare disorders caused by mutations. What are mutations? Sudden change in the gene structure. So the changes will be all of a sudden taking place, and that are called mutations. So SID is a group of rare disorders caused by mutations in different genes involved in the development and function of infection fighting immune cells. So here the question that is involved is infection fighting immune cells. Infants with SID appear healthy at birth but are highly susceptible to severe infections, and the condition is sometimes fatal, usually within the first year or second year of the life, unless infants receive immune restoring treatments such as transplants of blood, forming stem cells, gene therapy or enzyme therapy. More than 80% of SID patients do not have family history of the condition. However, development of newborn screening test has made it possible to detect SID before symptoms appear, helping ensure that affected infants receive self-saving treatments. More than a dozen genes have been implicated in SID, but gene defects are unknown in approximately 15% of the newborn screened SID in patients. According to an NIH-funded study, most often SID is inherited in an autosomal recessive pattern. So it is autosomal recessive, and in which both copies of a particular gene, one inherited from the mother and one from the father, contain defects. So both are contributing. The best known form of autosomal recessive SID is caused by ADA deficiency. That is what we said, ADA deficiency, in which infants lack the ADA enzyme, and this ADA enzyme is very much necessary for the survival of our own soldiers, namely T cells. X-linked SID, which is caused by mutations in a gene on the X chromosome, primarily affects male infants. Boys with this type of SID have white blood cells that grow and develop abnormally. As a consequence, they have low numbers of T cells and natural killer cells, and their B cells do not function. Imagine the fate when T cells and B cells do not function. So symptoms of SID occur in infancy and include serious or life-threatening infections, which may result in causing pneumonia, lung disease, chronic diarrhea, and candida, yeast infections of the mouth, and diarrhea, and pneumonia, again lung diseases caused by the fungus pneumocystis. So these are all the symptoms of a SID, and as we said, it is highly susceptible to severe infections. So SID is caused due to defect in the gene for the enzyme adenosine deaminase. So it is a hereditary disorder caused due to deletion of gene for adenosine deaminase, and the deficiency can be cured by bone marrow transplantation. However, in others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But in both the approaches, the patients are not completely cured. This is the problem. However, if the isolated gene from the bone marrow cells producing ADA is introduced into the cells at an early embryonic stage, see, this is important, early embryonic stages, it can be a permanent cure. That is the greatness of biotechnology. In case of many pathogens, a gene encoding a critical antigen has been isolated and expressed in bacteria or animals, and the recombinant protein so produced is used as vaccine. Such vaccines are called recombinant vaccines. A constituent polypeptide, in case of heteromultimeric proteins, or a small part of polypeptide may be used as a vaccine. Such vaccines are called subunit vaccines. So, enzyme adenosine deaminase is very crucial for immune function. In gene therapy, what is happening? Our WBC, lymphocytes from blood of the patients are isolated and grown in culture medium. So functional ADA and CDA introduced into lymphocytes through retroviral vector. This is the thing, and returned to the patients. Since lymphocytes have short lifespan, genetically engineered lymphocytes are periodically introduced into the patients. So what is important? Genetically engineered. But if gene isolate from bone marrow cells, producing ADA is introduced into cells in embryonic stage. Just now I said, what is the condition? It is embryonic. And once that is done, that embryo will not have any problem when it grows up to adult. That's a permanent cure. So how exactly it is done? This is how it is done. First, cells harvested from patient, and then in lab, virus altered, so it cannot reproduce. This is done. And a gene is inserted into the virus. Of course, here we are talking of gene means gene of interest. That's inserted. So plasmid. Altered virus mixed with the patient cells. Number 4. And cells become genetically altered because of the insertion. And altered cells are injected into patient's body. Now the condition is altered cells. And once altered cells are introduced into the patient body, that produces desired proteins. And once desired proteins are present, there is no problem for the production of the enzyme. What is the enzyme? ADA. So once the ADA is produced, then there is no problem. That is why adenosine deaminase is, once it is produced, then there is no disease and the baby can survive. And this happens only through gene therapy, thanks to it. And when we are doing this through gene therapy, as I repeatedly said, it's a permanent cure. But through medicines, through injections, it's not a permanent cure. Hence, thanks to gene therapy, we'll discuss next molecular diagnosis in our next session.